Hi everyone, and today I'm going to present topic next generation sequencing proteomics. So what we're going to talk? Of course, this presentation going to stand a bit as outlier from the most of the presentation, but I think it's very important to highlight how can you use very huge and quickly growing topic as next generation sequencing, which now even can like in routine way sequence the single cells on transcriptomics and another omics. So how can we use this information in proteomics? Is actually to improve proteomics result or to find something else or to answer for some new biological questions. So yeah, let's start from the very simple thing. So if you look how the central dogma works, so there's a DNA, RNA, and proteins, but on each level you have different degrees of freedom. For example, in DNA, you have copy number variation, you have mutations, you have epigenetics, for example, methylation. On RNA level, you have expression splicing, modification, secondary structure, also localization. And you see you kind of all these degrees of freedom are multiplied on all degrees of freedom which you had on DNA level. And of course, proteomics, proteins kind of as an integrative function over the all previous levels. And on top of that, there's a variability and degrees of freedom on expression, interaction, cell localization, post-translation modification. And that's basically why we're studying all proteomics method, because yeah, this is the single way for now to do it in macro on omics, truly omics level to study all those things. So, but from another point of view, let's look how NGS evolved for now. And from this perspective, we see that NGS can target many steps of this pyramid, including, of course, chromatin IP. So we can study epigenetic marks. We can study even chromatin structure using high-C generations. We can study DNA binding proteins. And all this is possible now. And thanks to the big projects, all this data accessible and everybody can use that. From another point of view, there's also many projects also on patient samples which study. Oh, actually, I said that I'm going to do that, and I'm going to put laser point. That's going to be really nice. So whole genome and exome sequencing allows to find out mutations, like driving mutations and passenger mutations in cancer samples, and really try to also helps to understand genetic population and etc. So, but from another point of view, there's also expression information which we can retrieve, can we get from NGS, and this is mRNA sequencing, and that's not just expression level, but we also using the current development of NGS, we can study isoforms. We can also study how mutations are passing from one step to another. We can study even second structure of the RNA, which is actually a big thing, and we can study even editing, post-transcriptional editing. And also, I think the closest omics up to now, which closest NGS omics, which coming to the proteomics, which kind of conference about, is ribosome profiling. In ribosome profiling, ribosomes basically covering approximately 30 nucleotides. And what you can do, you can once freeze them in the place where you're translating, digest all RNA, and extract the small RNA, which is like a sausage in between, like in hot dog, where the bread is a ribosome, and you can take this sausage and you can basically sequence that, and by that you understand where a ribosome was sitting, how many of them were sitting, and how actively a certain transcript or isoform was translated, and that's really wonderful topic. Okay, cool, but one thing which is a very common topic is variant extraction, and as you may understand, there's some variants which are non-synonymous. That means that this variants can be can change and can alter protein sequence, amino acid sequence. And the question is, okay, but what if I would like to study that? Can I somehow integrate these mutations in the MaxQuant search? That could be useful for certain application, and we're going to discuss for which application is especially going to be useful. But let's first of all, let's look how it works in general. So in general, how we are dealing with mutations, variants in MaxQuant So it consists of 3 steps. First step is variant extraction, when we're taking transcriptomic genomics data as like NGS data, BAM files usually, and extracting mutations which very relax calling rules. So we are really taking a lot of false positive variants. But if we stop on that and we start making some conclusions out of the mutations which we found, variants which we found, maybe it would be really not sufficient enough. But the thing is that we can allow so many false positives because we are filtering all these variants through the proteomics lately. And we're doing this using MaxQuant. So we are taking from one side FASTA file, which contains information about mutations which are present, and we are taking proteomics files, which is like any experiment from patient or from cell line, and we are merging variants to the database. We are suggesting to match some peptides with variants, and we are at the end we are finding actually a portion of the peptides which with variants, and we can do some downstream analysis. And as you see, this data analysis is can go forever. It's basically while true, and but there's a broad application for such type of data, and I would like to highlight a few of them. And for example, variant confirmation prioritization, which actually can be very powerful. But from another point of view, there's actually very truly biological questions. For example, how mutation influencing on the stability of the protein? Does it influence the stability of RNA? And using proteomics, we kind of actually can answer for the first question. Is it actually influencing on how on stability on protein, etc.? But there's actually even more, and I will describe a few more examples how we used in our lab. Cool, but I have to kind of say that it's very easy to use, so it's going to be some small publication, some small protocol describing how to proceed step by step, but this is already in the MaxQuant software. If you go to tools, you can find variation extraction, and you find out the GUI where you can specify all your NGS data, also way to save information, and where you have to upload FASTA file with a genome itself and annotation. It's actually very easy to use, and this protocol will be very useful when it's published soon. Okay, but let's show some examples. How can we use this data? So in order to show the actual usefulness of and check how this pipeline works, we took the data from this paper where the researchers studied HeLa for the first time with very high depth. And how did we reach this high depth? With 2 parameters. So two methods. First of all, we used a very very generous fractionation. So each sample was fractioned for, I remember, like 34 fractions. And on top of that, we used different proteases. So imagine, so we're taking, let's say, HeLa cell line. We are digesting with chymotrypsin, with gluC, with lyC, trypsin, and this is all happens in parallel, not in one tube, of course. And each of this sample we're fractionating for 39 fractions. We are getting really very deep proteome, but the question is how many mutations, how many variants we can find which are non-synonymous with different proteases. So as you see, trypsin is still the leader. So we see approximately 715 variants, not like peptides, variants which we can be confirmed using proteomics with lyC 608, etc. But how many variants are uniquely identified thanks to trypsin? And we see it's 191. But that's actually also, but how many unique identifications we have for lyC? 168. And we see also all combinations, and for example, this hero 72 variants which we found using all proteases. So what's this picture tells us? So first of all, that's using trypsin, just trypsin, it can be actually not so advantageous to find many variants. So, and it's pretty clear, if you want to go for a really high coverage proteomics, then using different proteases is the way to go. That's pretty much a fact. But we also see that it's really advantageous, and we see way more variants thanks to the usage of different proteases. Okay, so, but also let's now look at all variants which we detected from the perspective that's okay, we have reference amino acid and we have alternative amino acids. And first of question, how many actually we detect transversion, like variations from one amino acid to another amino acid? And this this information can be found from the size of the size of the circle, and they see that's totally not uniform. And there's probably two reasons. So first reason, that's all amino acids, we don't have a uniform distribution of frequencies of different amino acids, that's for sure. But also there's a physical chemical properties of amino acids. So as a result, some changes are really dramatic in terms of evolution, in terms of stability of protein. That's why we filter out. So also the third actually fact that there's a genetic code, which also puts some restrictions on the what is easy to change, what is not easy to change. But it's a first dimension. But the second dimension, which we also can look, we can ask, but how many of 100%, let's say we see from transcriptomics data or from genomics data, we see 100% non-synonymous mutations. And the question is, out of this 100 non-synonymous mutations, how many actually we are detecting using proteomics? And this is what this color is about. Of course, it would be wonderful if everything would be red, but that's not actually what's happening usually, because we're still not reaching 100% coverage. But we can see that in average, it's around 30–40%. And that's what we're working right now with, and that's actually already informative. So we are really detecting 40% of variants which are translated from transcriptomic level to proteomics level. So yeah, cool. So what can we do more? So yeah, so as I said, there's a few actually very cool applications which we can explore. And for example, one of the application, allele specific expression. So I have to say, here we are discussing allele specific expression due to the copy number variation changes. So, and of course, maybe many of you knows that HeLa cell line is really famous for that. So we have HeLa cell line, most of the chromosomes actually living in the state of copy number 3, and as a result, it could happen that one chromosome have a variant, and this variant, let's say, have a G in the third position of the codon, and this G results in these amino acids. But into the rest of the chromosomes, we have a C in this position, and this results in non-amino acids. So the question is, if we see this split 1:2 reference to alternative, will we see the same split on proteomics level if we compare peptides, which are coming from with one amino acid or another amino acid? That's actually, it's not like totally allele specific. I mean, it's like, let's say, copy number variation driven allele specific expression. So, and how can we do that? We can actually look at this from two perspectives. From RNA level, we can just divide a number of reference reads which support reference nucleotide and alternative nucleotide, number of fragments, number of reads which support alternative nucleotide. So also from proteomics level, we can sum up intensities of peptides through different fractions, through different charges, through different modifications, which support one amino acid as a result, and one mutation, reference compared to the alternative. And now we can basically plot something like that. So we see that because we have replicates, we can really make a distribution, we can estimate standard deviation, and we make a correlation matrix. So as a result, we can really plot 2D Gaussian distribution. And we know from genomics information, we know the copy number of each position in genome. And we know that this mutation is happening in copy number 3. Cool. Okay, but now let's look how this ratio of reference to alternative looks like from transcriptomic point. And we see that this cloud is around -1. That would means that it's logarithmic scale. As a result, it's 1:2 ratio. So that means that we have one chromosome with reference variant and two chromosomes with alternative variant. And the cool thing about this study is that we actually can also look at how peptides translate this like changes. So, and we also see that in this case, peptides intensities of peptides ratio is actually also around of -1, and we see that proteomics also totally agrees with transcriptomics and with genomic information. Yeah, but there's another cases. I told you the HeLa cell line is really crazy cell line from the perspective of the copy numbers, and there's let's say copy number 4, and that can be many different combinations, like combination 1 to 3, 2 to 2, and 3 to 1, and as a result, we actually do see distribution of this variant around of 0.00. That's what means that we have 2 to 2. So we basically have 2 chromosomes with reference and 2 chromosomes with alternative. So, and now if we take all plethora of the variants which we see in the HeLa, and we know what's the copy number we are looking at, we can see some distribution like that. This is pretty messy, but let's look at the copy number 3. We're just taking the variants which are coming from the chromosome positions with copy number 3, and we see some diagonal. And this is pretty much what we expect to see. So we see some heap, like let's say the mode on the -1, and we see another mode on 1 point. That's what basically means that here we have 1 to 2 reference to alternative, and here we have 2 to 1 reference to alternative ratio. But also you can see this on proteomics level, the standard deviation is nothing comparable to RNA sequence level. And that's probably we can blame physical chemical properties of the peptides. That's maybe mutations or variants basically is bringing some physical chemical changes to the peptide. But this is something have to be explored. And honestly, I can predict that some technologies of very targeted approaches, for example, like PRM, could be really useful to do it really precise in concentration-wise, and that's, I think, the future of such type of studies. But also, again, we discussed here copy number-driven allele-specific expression, but allele-specific expression happens actually even on when you have 2 copy numbers, and we have one allele which is more like more active compared to another one, and this can be also studied, I'm quite sure, with this tool and with together with PRM. Some advanced proteomics. Okay, cool. But another approach, another application which I would like to talk today about, this is immunopeptides. And I was involved myself in the project, which was using the variants which appear on immunopeptides, and actually very grateful to, very happy to discuss it today. So, but as I said, you remember that previously we were able to analyze variants. We were able to find variants when we use database which constructed based on the different proteases. But we can also use unspecific search. We can say that now we'd like to search for the all peptides length of 8 until 11, both ends included, and all peptides there which do have mutation would be interesting for us. And this is actually really, it's usually this immunopeptides, I will describe what's actually we're doing, but for now, this let's put a constant that's this peptides usually a length of 9 amino acids, and we do have very specific patterns. We do have anchor amino acids in certain positions because it helps to load this peptide on the surface of the cell and present to immunity. So. But this study, actually, I really recommend to check this study. This is actually a very nice article, what people did there. So it's already follow up on another article. But so we just, maybe each cell line of our body is expressing 6 versions of HLA proteins. HLA proteins is basically the glove which is presenting 9 amino acids peptide, usually 9 amino acids peptide, on the surface of the cell. It's presenting to immunity, for immunity to recognize it, and maybe kill cell because it's made dangerous, and we discuss why it can be dangerous. But again, if you take a patient sample, on the surface of this patient sample is going to be 6 HLA proteins, and each of this HLA protein do have certain specificity. It's like certain amino acids, certain positions, and all these things. But this study focused on the actually engineered cell line. In this cell line, we removed all HLA proteins from the genome, and next we started sorting one HLA one by one. So, for example, first cell line was constructed after we removed all HLA proteins, and we inserted into the genome this HLA A0201. Okay, cool. And now we are taking all peptides which are present on this cell line, and we see such pattern. That's very specific pattern for this HLA protein. But next, we are taking again the cell line with no HLA proteins at all. We are inserting another HLA A3303, and we are taking all peptides from the surface which are present thanks to these proteins, and we are sequencing that using the proteomics. And it's really possible, and you see we're detecting some reasonable amount of peptides. And we can do it with many HLA proteins. And I have to really tell you that HLA protein is one of the most variable protein in the human genome. And so there's no surprise. This study, I think, has already hundreds of the HLA proteins. And this really represents variability in the human proteome, in the human population. But now let's introduce. We know the cell line. We know the genome of the cell line. We know the transcription of the cell line. We have transcriptomic data of the cell line. That means that we can take the mutations, variants, which we see from data, and we can insert it to the database and search against this variants. Can we find them or not? And as a result, and also you can see we have really perpendicular checkup. It's like sanity check. Do these peptides, which we find using the variants, do we kind of reproduce the pattern which we see for the reference peptide? This is like really very simple but very elegant sanity check. So, and we see it's actually true. No matter which HLA we take, we see that this pattern is really reproducible. But this peptide, even it's a few, yes, it's few, but even though we see the pattern, we see that this pattern is nicely reproduced. Cool, but now it's time for some nice stories. So we have in Max Planck Institute, we have a RIP seminar. And once I was presenting there, and they asked me to, and we were discussing to make a poster which we bring around the institute and ask people to join for this RIP seminar. And the first version of this poster was like that: cancer T cells and 2 smoking immunopeptides. So I think the administration of the institute decided that's not really elegant poster, so it was not accepted. It was rejected. But okay, let's talk about how T cells recognize criminals. So as I said, each cell in our body have a protein called MHC1 or HLA is basically the same. And this the single purpose of this protein is to represent peptides on the surface. Like basically it's just taking, it's really crazy system insights, but there's some immunoproteasome which is by chance digesting proteins by chance. And the peptides, which are approximately 9 amino acids length, which are suitable for this MHC pattern, it's loaded on MHC, and at the end it's represented on the surface of the cell. So, and the next police, our immune system T cell is coming and is really trying to touch this peptide. And if it's a peptide which is not host peptide, which is unknown for immune system, I mean, it's tricky, but then immune system is triggered and getting pretty aggressive. So for example, if the peptide with non-synonymous mutations appeared on the surface, some immune cells can recognize it as a non-host, and it will push the cell to die. Why? Because it's potentially is a cancer cell, so it really accumulates mutations, and probably it's worth to kill it. But another, and which is actually a very frequent case, when we have a cell and it's infected with virus particles, and this virus is sequencing on proteins, and these proteins also can be by chance digested with the immunoprotease, and also by chance can be represented on the surface. And as a result, some immune cell can recognize it and sell the signals to the cell to please to die, because otherwise whole body gonna be infected. So, but in this study, which we did in our group, we were focused on the cancer story. And why it's so interesting to study. The reason, because if we would be able systematically, robustly identify mutated peptides and mutations which appear during the cancer development, then we would be able to synthesize this peptide and actually really train immune system against this peptide. This is basically like, like a dog trained against a certain smell, and this smell is this peptide, and this dog gonna go around the body and find out whatever is presenting this peptide, which is gonna be cancer plus metastasis. And this is really, sounds like really good method to use for a cancer treatment. So there's a lot, of course, but that's a matter of this research. Let's study that. So, but what we did in our study, this is exactly the publication if you're interested in. So in this study, we checked the melanoma patients. So we took from them blood as a representative of the reference, as a representative of the basis, a basic proteome, so the proteome which was not altered during the cancer development. And also we studied a tumor genome using whole exome sequencing. So, and we were hoping to find mutations which are specific for the tumor, but are not present in the blood. So this is exactly mutations, as you may understand, which are cancer specific, appear during the cancer development. So as a result, we're finding subset of these mutations are going to be non-synonymous, and we're including actually all mutations which we find into the search. And lately, we are checking out who is actually non-synonymous mutations who are tumor specific, and we're using exactly that mutations and peptides which have that for further studies. But from proteomic side, we're taking the same tumor cells. We are taking the HLA proteins from the surface. It can be different approaches, but like at the end of the day, we're just taking the peptides from these gloves, and we are analyzing using mass spectrometry. So lately, we're finding peptides with somatic mutations, means that's cancer-specific mutations, and these peptides potentially can serve as a vaccine, as a personalized vaccine against the cancer. But in this study, we just checked, it made the immunogenicity assessment that there's actually a reaction of this on these peptides in the blood, but potentially it can be actually really nicely used with together with therapies, with chemotherapy or STLA4, PD-L1 blockage therapy. So we actually pretty, I think there's a lot of opportunity in this direction. But from another point of view, why we study melanoma? But this is a very simple question, because if you look at this slide, what does it mean? There's a number of somatic mutations prevalence. So it basically shows you how many mutations, new mutations, cancer-specific mutations you have per megabase, per genome length. And we see that melanoma is top one. In average, samples from melanoma do have really a lot of mutations. And there's a reason for that. It's a reparation system is broken, and as a result, cells are really accumulating a lot of mutations. But actually, that's why we select that, because we're going to have a lot of mutations. Some of them going to be non-synonymous, and as a result, we're going to probably find out some mutations which are using the proteomics data. And actually, what we did, and I have to say, we were finding really few, really 1, 2, 3 peptides per patient, and we were using an immunogenicity assessment. But that's not what I would like to highlight today. What I would like to highlight today, that's before most of the studies, we were focused just on NGS. So we were taking PBMC, so blood, and we were taking tumor samples, and we were analyzing with whole genome sequencing or RNA sequence data or whole genome sequencing data, and we were finding these mutations. What we do next with these mutations? The mutations which are cancer specific, we are taking all possible 9 amino acids around of this mutation, and we were predicting probability of this mutation to be present on the surface. So this was totally in silico prediction, and let's see how approximately it works. So this is in silico prediction which was done with new epitopes, NetMHC program. So this is how usually it looks like. So on the first graph, we have predicted affinity against certain HLA protein. And the low, like 0 affinity means it's very strongly binds to this HLA protein. The high value means very weakly binds to that. And x-axis is just rank of this value. So the 0 is the strong binder, 1000 is a weak binder. So you see, there's not just few mutations, few peptides which you can find using genomic data. So you see this gray dots, it's all mutations which passing the limits 500 nanomoles, which have somatic nature, which we find in these patients. So there's really a lot. And if you want to check each of them one by one, probably you start from the most strong binders and you go to the weak binders. But this is a really very elaborative process. It's require a lot of work, etc. So, but what we can do, we can actually prioritize mutations using proteomics. So we found a few variants. We found a few peptides which contain these variants, and we see them here. So this is SYTL4, and you see if you will go there and you will find it. If you're really using the old genomics level of genomics experiment, you will go one by one, check each peptide, and just after 70th check, you go to the 18th check, and these peptides we're going to be actually present and actually very strong binder and probably immunogenic. So what I try to highlight here, that's proteomics here. Proteomics serves a really good role. It's actually really confirms that this peptide exists on the surface, and that's exists in the significant amounts to detect it, and that's can be nicely used for the future of development of the patient-specific vaccines against cancer. I really think that there's a great future for this experiments, but let's move on. So that's the second poster suggestion, which was, as you may understand, realize it was also rejected. But overall, this was one flew over immunopeptidomics, and I hope you like it, and this was not really crazy. So, okay, and for the rest of time of my presentation, I would like to also show you what you can also do with your NGS data in the MaxQuant Perseus environment. And for example, you can also analyze gene expression. You can make gene expression analysis in Perseus. And more specifically, let's just discuss how it's usually done in the NGS world. So in the NGS world, you have Illumina machine from Illumina, not from Thermo. And there's a machine generates FastQ files. So it's actually also funny to compare how NGS world develops compared to the proteomics world, and you actually very quickly understand in NGS world you're getting sequences as output from machine, but in proteomics you're getting a spectrum from machines, which are way more complicated, require interpretation, very robust statistical interpretation, and that's why actually proteomics is a cool field, because it's complicated. So, but you can take this raw files from instrument, which is usually present as in FastQ file, and what you have to do first, you have to really match it back. You have to understand where this fragment is coming from the genome, and this is called alignment to genome. And usually you're getting files, and it's usually really several gigabyte files, and this is usually BAM files, files which do have information about where is the certain fragment aligned to the genome. And having this information in hand, it's very easy to estimate gene expression, kind of estimate gene expression. You just can quantify how many fragments you have per gene, and this is going to be a gene expression. Of course, you may ask me about, but what's about isoform expression? Yeah, so can you actually identify isoform expression from that? Yes and no, but let's be honest, it's a very complicated thing to do. So we also have some progress in this direction. And today, plenary speaker Joshua Kuhn gonna discuss that in more details. But okay, but let's see how it's actually the whole field is developed now. So it's actually a very open field. So we have also some kind of pride. But for the genomic data, it's for example, European and Nucleotide Archive. There's a GeoSRA database, and there's also ENCODE projects and many more. Which are actually depositing not just raw files, but already aligned data. And this is actually very useful because you can just download these files. For example, in ENCODE, you can find almost for all cell line, you can find transcriptomic data, and you can just take them and estimate gene expression, how many different genes expressed, how many fragments, and you can compare with proteomics. That can be actually very useful. And because that's may be useful and can be cool to have like in quick usage, so we kind of really applied, we made accessible that this functionality starting from the alignment to the gene expression in Perseus. So it's approximately looks like that. So there's one of the activity in tutorials for the Perseus probably will show that, but there's one activity, upload activity, and it's called next generation sequence data upload. And there you can just specify the file, which basically the bound files of the alignment. You can specify many parameters, including genome annotation. Genome annotation, like where each gene stands in positions. So you can, there's many parameters here, but there's a number of threads. You can make it multi-parallel, and as a result, you're getting results, and actually you're getting results really quickly. So we'll discuss that. But before that, let's just introduce a few things. You see there's a strand specificity parameter here, and by default, it's written as not stranded. And that basically means that all fragments which we have where don't have any directionality, we are directed 50% one direction, 50% another direction, and but the current modern like NGS transcriptomics standards such that you can easily make stranded library and sequences in a stranded way, preserve this information, and antisense. So the stranded but in another direction. So, but honestly, almost 99% of the cases which I see, just non-stranded would be totally enough. Just if you don't know which strand, just use non-stranded, just use default, and there's not going to be any problems. So, but of course, we are not the first people who are doing that, and there's a lot of development in this direction going on. And one of the tool which used a lot in NGS world is HTC count, and this is basically Python framework around of the C code, which is trying to access the one file in fast manner. But we can find out that we just can take a few samples which you can access from the geo database. We can analyze with a single thread, and I just analyzed chromosome 1 just for simplicity, and I found out that there's a really nice correlation, a nice correlation between replicates within the software, and there's almost the same results between Perseus and HTC account between the same replicates as you see here, and that's pretty nice. So, but one of the biggest advantage I think to use Perseus, that it's extremely quick. So because, and one conference I think Matthias Mann asked why it's so quick. I think the main reason because we're using the same platform. We're using everywhere C# from reading a BAM file and analyzing all these things, and we are doing it very efficiently. And that's why it's so quick. As a result, you see, so we can easily take data for your specific cell line, which you're studying from, like, for example, ENCODE or SRA database, and you can easily and quickly find out expression of different genes and follow up and make follow up with this. Cool. Okay, but how can you use that? And the few things which let's describe it, just a few stories. How can you use that? So one of the things, as we are coming from the proteomic world, we can actually compare our results just back to back. We can compare, okay, we have proteomics data, we see IBEC. It's actually IBEC is really the let's say proteomics alternative for genomics RPKM, transcriptomics RPKM. That's why it's used here. We're really very well comparable, and we're just taking the comparing IBEC versus RPKM. It's just normalized value of the transcription expression, and we can compare between what? We can compare between three omics: proteomics. Let's start from beginning, from transcriptomics, translatomics. So there's exactly the ribosomes, how many ribosomes we have per gene, and we can compare with proteomics. And so the first question which we may ask, like, with whom proteomics correlates best? And as you see, this is comparison between transcriptomics and proteomics, and this comparison between translatomics, ribosome profiling, and proteomics. And we see that proteomics is way better correlates with translatomics. And I think it makes sense because, you know, there's not so many degrees of freedom between when translation is actually already happening and there's a protein exists. Of course, there's a destruction of the proteins. There's different modifications. But anyway, So there's a way less differences, way better correlation between translatomics and proteomics compared to transcriptomics and proteomics. So another story which looks like that, together with Dan, we analyzed one data set. So we somehow destroyed a chaperone EMC2, which helps proteins to get shape. If you are destroying this protein EMC2 and compare to this to control, now what can we do? We can compare each dot is going to be, let's say, gene, and we would like to compare how this gene behaves on translational level. Is the translation is increased or decreased, and how it behaves on proteomics level. And this is actually, we have several groups, and we made a big follow-up on each group. But let's, for example, focus on these green dots, green-blue dots. As you see, for that proteins, translation that haven't changed at all. But for these proteins, what changed? Protein abundance, which we see with proteomics. And that's actually really cool. This means that these proteins do express, do translated, but while there's no chaperone which will bring them to the shape, we're really directed to the degradation. And that's actually a very beautiful story, I think. Cool, but overall, you can analyze cell lines, and you can do it very quickly. For example, analyze this many replicates of different cell lines from the ENCODE project for 40 minutes on my computer. So it's pretty reasonable and pretty quick. So I would like to thank all my group once more. And also I would like to say a big thank to Max, who helped me a lot with many activities which I described today, including the variant activity and actually writing publication.